हाय एवरीवन। वेलकम बैक टू माय चैनल। यार, हम सब लाइफ में कभी ना कभी बीमार हुए और हम सब ने ये मेडिसिन लिए है। बट हैव यू एवर थॉट फ्रॉम वेयर दिस एक्सपायरी डेट केम? डोंट वरी माय फ्रेंड, टुडे इन दिस वीडियो आई विल कवर दैट टॉपिक. My name is Simon. I did my master's in pharmaceutical analysis and currently I'm working as a senior research scientist in one of the German multinational company. So without wasting further time, let's start the video. So the topic of the today's presentation is important terminologies to understand the ICH quality guideline. I call it important because these terminologies, they are simple, but they are so important that if you don't understand these terminologies clearly, it is very difficult to understand the ICH quality guidelines. And that is why a dedicated video for this terminologies. So let's see the content of this presentation. So first I'll explain what is drug substance, excipients and products, followed by what is stability study, how many types of stability studies are there and what is the importance of doing this, followed by what is force degradation studies or stress testing, why we are doing it, then release spec, shelf-life spec or specification in general, then what are the types of batches that in pharmaceutical industry they are basically taken. Okay, so let's start. So first we'll discuss about the drug substance, excipients and drug product. So drug substance, you can say it is nothing but, okay, first of all, the definitions that you can see on the screen, that those are taken from the ICH. So they are the official definition and uh, you don't need to go through the definition. I am here to explain it to you. So drug substance means it is nothing but the raw material of any medicine. In pharmaceutical industry, we call it the active pharmaceutical ingredient. So in simple terms, this is the material or molecule which is responsible for curing your disease. For example, let's take this one. I'm having a fever. I took paracetamol tablet and my fever got reduced. Is it the tablet which has worked or is it the paracetamol? The answer is it is the paracetamol which has worked. Okay, I could have taken paracetamol in any form. I could have taken it as a syrup or I could have taken it as an injection, but I chosen as a tablet. So it is the paracetamol which has reduced my fever, not the tablet. Always keep in mind. So it is the drug substance which is the active pharmaceutical ingredient, and whenever the drug substance it is mixed with the excipients and then it form the final product. So excipients are nothing but whatever the other thing you mixed with the drug substance to make the final product that becomes your excipient, and the final product is the final product, right? That is also known as drug product. So if I have to give you a simple diagram, you can see on the screen that drug substance it is mixed with excipients and it forms the final. Now let me give a very simple example from your day-to-day life so you can easily understand. Suppose you want to make a paneer item, for example paneer curry you want to make. So you want to market and you bought paneer. So now think of it paneer as a drug substance. Okay, now you mix masalas with it, all the sugar and salt, water, and you make the final paneer curry, the dish. So whatever you add in between like masala, sugar, salt, water, that are your excipients, and the final, that paneer curry, that becomes your drug product. You can think of it like that. Okay, and the final product can be anything. You can prepare paneer curry, paneer um, um, chili paneer or sahi paneer, so that can change, but the paneer, the raw material, that would be constant. So similar here, the drug substance mixed with excipients and they form drug product. So I hope now it is very much clear to you. Okay, so now we'll understand the stability studies, and it is very important when it comes to pharmaceutical industry. Now remember I told you in the introduction video that from where these expiry dates come. My dear friends, the expiry dates, they are basically determined with the help of the stability studies. So what the pharmaceutical companies do, that whenever they manufacture these medicines, after manufacturing, they generally store it in a stability chamber. So one of the kind of stability chamber, you can see it on the screen. And this stability chamber, they have predetermined temperature and humidity set. Okay. So they load, after manufacturing, they load their tablet into that chamber, and from time to time they pick out those samples and they test it, and they check it how the results are coming and up to what time the results are passing against a set test. Okay, and then they tell you that it is stable for 3 years or it is stable for 2 years. So what they do, they um, load their samples into the different stability chambers, and they have some of the chambers, they are long term. Okay, that means the temperature and the humidity, they are kept at recommended storage, and some of the chambers they have accelerated, that means some of the chambers have higher temperature and higher humidity, which can mimic the long term result in a short period of time. Now long term testing is basically the recommended storage condition. For example, this is the medicine, right? Now, if I look back on the tablet, it is written that store at temperature not exceeding 30 degree and protect from light and moisture. So my friend, the recommended storage condition that stored at 30 degree, whatever it is written, generally it is the long-term testing. Okay, and suppose whatever the suppose the result that is going to come after 2 years or 3 years, if you are going to predict that one in a short period of time, what you will do, you will increase the temperature and humidity for a short period of time, and that is nothing but the accelerated testing. Okay, and there is another one term called intermediate testing, and it is basically the temperature and humidity which is in between both long term and accelerated. So this is just an overview of long-term, accelerated, and intermediate. There are many such terms and conditions are there, like what temperature you have to follow, for how long you have to do, what time point you have to pick your samples and test. That all I am going to cover in my from my next video. Those are going to be very detailed video. Now, but as of now, this level of understanding, I think it is very important. Okay. So now we'll go to the next slide. Stress testing or force degradation studies. Now remember I told you that the accelerated testing is a little bit higher, okay, than the long-term testing, or you can say a little bit harsh. Now the force stress testing or force degradation studies, they are far more harsher than any long-term, any accelerated testing, and these are generally carried out in the development batches, okay, or development stage or R&D stage, where there is a they are working on the molecule or formulation, okay, or analytical method development. Now, why these studies are very important? In my first video regarding in the ICH, basics of ICH, I told you that in 1950s or 1960s, there was a great mishappening that happened due to the drug substance. Many children got died. So safety in pharmaceutical industry, safety is very important. Whether your drug working or not, that takes the second role, but the first thing is whether it is safe or not. So stress studies is kind of related to that. Okay, so what it says do, in these studies we intentionally degrade our drug. I again repeat, we intentionally degrade a drug or spoil our drug. Okay, and then you check what are the molecules that it is forming, how much it is forming, is it toxic or not? If it is toxic, how can we reduce it? What are the formulation changes we can do so that it won't form? So this is the stress testing. So there are basically 6 types of exposure that we do: acid exposure, base exposure, peroxide exposure, thermal exposure, humidity exposure, and photo exposure. So basically we took our samples and we expose them to this situation, and then we see how they are reacting. Okay, what are the molecules they are forming, and on the basis of that we optimize everything. Now, to understand it more clearly, first thing a force degradation studies helps to do is to establish degradation pathway. Now let me give you an idea so that you understand it better. So suppose I took my drug product and I expose it to acid, base, peroxide, thermal, humidity and light, as you can see on the screen, right? And suppose upon exposure it to the acidic condition, it forms a molecule called A. Then upon base, it forms a molecule called B. And when you expose it to the peroxides, it forms A, B, and a new molecule called C, and three together. Then you expose it to thermal, it forms D. And similarly, in case of humidity, it forms D and E, and in case of light, it forms F. Okay, so these are the new molecules, or we can say impurity. In pharmaceutical in the industry, we call it impurities. These are not the impurities because these are the molecules which has make your product impure, right? So that is, we call it impurities. Now suppose we take our sample as an industry, and then we load it to a stability chamber, and then down the line, after 2 years, when you took the sample, which is suddenly we found that there is a new impurity, or this molecule is coming, that is D plus E, right? Now we might be thinking that, are there is a D plus E, these two molecules are coming, so at what degradations these molecules came? Then we go back to our test studies or force degradation studies, and then you link, are these are the molecules which came in humidity, so there might be a humidity degradation. That means my packaging system won't be strong enough to prevent moisture there, or somewhere moisture is penetrating in my sample, or the tablet I have that might be absorbing moisture from its nature. So there have to be some formulation, the uh, optimization that needs to be done, and that is how the industry thinks, that is how the R&D is being done. So I hope now we understand what is the importance of stability studies. Okay, so this is the first, establishing degradation pathway. Now second, these are very important to develop and validate an analytical method. Now when you expose your samples to all these degradation conditions and there is a formation of impurity, and now you develop your analytical method on the basis of all the impurities. Suppose there are formation of 10 impurities in all the degradation, 10 impurities, and you develop your method, analytical method, such a way that it can detect all the 10 impurities. And suppose down the line, after 2 years or after 1 year, there is a formation of one impurity. By the time the impurity will form, your analytical method is capable enough to detect that one, right? So if you didn't develop your method on the basis of those 10 impurities, it could have never detect that molecule after 1 year, after 2 years that it's formed. And that is why whenever you develop your analytical method, that is an impurity detecting method, or RS method we call it should detect all the impurities, and that method is generally developed on the result, on the basis of the stress studies. Okay, so that is very important. I hope you now you understand that one. Third point, obviously, the formulation development that I already explained. And finally, is the packaging system optimization. Now, apart from these four, there are many other ways it can be utilized. You can see there is a pie chart and many other things are given. Now, please be note that this is just a basic introduction of force degradation studies. In the upcoming times, I'm going to take a detailed session on that, where I'll tell you how to predict from the molecular structure that which under which degradation is can go. Okay, what are the conditions of taken for this degradation studies, how for how long it is being done, everything, what are the regulatory requirements for that, everything I'll cover. But as of now, this much of basics is very important. Now this is also very important slide. What is specification, release specification, and self-class specification? These are the terminologies you will hear almost every day in a pharmaceutical industry. Okay, and without these terminologies, you will not understand ICH guideline. Now, specification in simple language, the definition you can see on the screen, that is the official ICH definition. Okay, in simple language, it is a document which is having all the test mentioned, and along with the expected results or appropriate results. So whenever you test your drug product in a pharmaceutical industry and check whether it is pass or fail, you basically test against that document, and the result, for example, there are 10 tests they are mentioned as you can see on the screen, test parameters, and there is a specification what is the value it should be. Okay, and they are followed by reference from where this test parameter came, whether it is in-house or whether it is taken from some other thing, that also we'll discuss. Okay, so this is basically test, and we tell either passing or failing. Now these specifications, there are two types. One is release and another one is shelf life specification. So in commercial scale, whenever the pharmaceutical company they manufacture medicines, just after manufacturing, they use one specification and they tested the product, and if it when it is passing, then they launch the product in the market. And when it is the market, then the testing again, it is being tested, okay, and then it is tested against the shelf life specification, okay. So please be note that the release specification, it generally it is more stringent than the shelf life specification. The result is simple. Suppose you make, again take paneer example. Suppose you today you prepare paneer, okay, and you take it, it is very hot and fresh, you took it, you find it very delicious, okay, and then you kept it room temperature for tomorrow or for one day, and day after tomorrow when you are going to take the same paneer, do straightforward will take it, or will take it or taste it for smell it whether it is good or bad. And if you find it is good, now that the smell is good, now taste is good, now I can take it. Similar thing happened to pharmaceutical industry. When you prepare the medicines, it is very fresh, so the result they should come close to the actual value whatever the label claim is there. So the specification is very tight, okay, the limit is very narrow at that time. But when you launch it to the market and there is a time like of 1 year 2 year it is going to be in the market, so in that period there might be some chances of degradation in your product. So in that cases the specification is little bit broader, the acceptance range is little bit broader. I hope you understand like what is release spec and shelf life spec. And remember, release spec are generally more stringent than shelf life spec. Now in this slide we'll discuss what is development batch, exhibit batch, pilot scale batch and commercial batch. So first understand what is a batch, then we'll go into these topics. So again, take some real life example. So suppose there is a family function is that is going in your home, that is going to happen in your home, and there are around 10 or 11 people who will come into your family for that function. And your parents, they have decided that this time they will prepare chicken biryani for them. And but the scenario is that in your home, uh, chicken biryani for 10 11 people it was never prepared before, okay. So the whatever the utensils you have, it is for 4 to 5 people, because in a family there are 4 to 5 people, you, your brother, your mom, your father, okay. So hardly 4 and 5 people. So why do you need utensils for 11 people, right? But this time you have to prepare it for 11 people. So what you will do, right? You will prepare in batches. First you will prepare for 5 people, then you will prepare for, or simultaneously you will prepare for another 5 people, so it becomes 2 batches. So this is it. In pharmaceutical industry, commercial scale, when you prepare very large scale, and suppose your container has a size limit, so you prepare in a batch by batch basis. For example, you have to prepare 20 lakhs tablet, but your capacity is to prepare 10 lakh tablet. So you prepare 10 and 10. So there 10 becomes 2 batches. So I hope now you understand what is a batch in general. Now, the development batch, we call it the R&D batch, okay, or also the technique, some industry it is called technical batch or engineering batch. So these batches are R&D batch or research and development batch, and these batches are taken to develop your formulation as well as analytical method, and these are lab scale, very small batch, probably 1 kg 2 kg or 1 liter 2 liter batch, okay, very small batch. Generally non-GMP batch. Non-GMP batch means that even if it fails or there is a failure, um, you don't have to take kappa or you don't have to take an events. Now what is kappa and events? These are nothing but the documented process which you have to take to maintain the quality of your manufacturing process. Now as it is a research and development, there is no need to go through this documental process every time there is a failure. So that is called non-GMP batches. So if there is a failure, you can redo the process. Okay, so this is a development batch, and generally it is a very small scale batch. Coming to exhibit batch, now this is a batch which is a little bit larger than the development batch. Okay, and this is GMP batch, and this is the batch which stability data we submit it to the regulatory agencies for the approval of the product. It is also known as primary batch and submission batch. Okay, in industry, when you start working, or if you are already a working professional, you must have heard about this exhibit batch term. Okay, now coming to the pilot scale batch. This is the batch which is actually the 1/10. General rule is 1/10 of a commercial batch. Okay, and this is fully representative of your commercial batch. That means whatever the vessels that you are going to use for a commercial batch, whatever the filter that you are going to use, whatever the manufacturing line that you are going to use, it is same. Just the thing is, it is 1/10 of your commercial batch. The exhibit batch, I remember I told you the exhibit batch, it is generally of a pilot scale batch. Okay, 1/10 of your commercial scale batch. And what is commercial batch? From the name itself, you can guess this is the batch which is used for market purpose, commercialization purpose. And this is the batch which is given to the humans. When it comes to the market, humans take it for human consumptions. You can also say it is this batch is also known as production batch or commitment batch also. So there are very many other names also for this. These are these batches. These are development, exhibit, pilot, and commercial. These are the standard names, but you will also hear the other terminologies also depending upon the industry to industry. So this is all about this presentation. uh, in my next video, I'll cover the ICH quality guideline in detail. Okay, region wise, what are the requirements, why we do, what are the things we do, and why you do, from a regulatory point of view and from a science point of view, obviously. And if you like the video, please subscribe, like, give your comments. If there is any improvement you feel that needs to be done, if there is any clarification needed from my side, please comment on the back. Well, I'll see you in my next video. Take care of yourself. Bye.